Okay, I'm going to talk about Lab 2 and its requirements. There's not that many adjustments to this, but there are just some kinds of terminology that I wanted to use a little, excuse me, differently. So in the same fashion that Lab 1 is about identifying 2 different types of carbohydrates, Lab 2 is about identifying proteins and identifying lipids. I wanted to point out that there are 2 separate sets of substances, unlike in Lab 1. In Lab 1, there was a single set of substances that you tested both with the Benedict's test and the iodine test. In Lab 2, there's 13 substances of one set for proteins. Buttermilk is actually almond milk if you wanted to change that on your hypothesis. And for lipids, there's 16 different substances. I would say that in your background, use the definition of protein from your textbook in Chapter 5 rather than the definition given on page 7 of the lab manual. You want to go to Chapter 5 for that. Really what they describe in the lab manual is a polypeptide, not really a protein. A polypeptide, it has to be folded into a particular shape that has some type of biological function for it to be then considered a protein. There's 2 different functional groups, special groups that have different characteristics on amino acids, which are the building blocks of protein. So here's your protein functional groups. Here is one called the amino group, nitrogen to 2 hydrogens, and this would be attached to carbon. But here's the amino functional group, and here is the carboxyl functional group. Draw that again for you. The carbon is attached by a double bond to oxygen. So that looks like a carbonyl. However, instead of adding another carbon or hydrogen, you add a hydroxyl group, and now no longer is this a carbonyl and this a hydroxyl. The whole thing is now called a carboxyl group. And it's completely different than either the carbonyl or the carboxyl group. In the test, the Benedict's test, we had a capital B and we had an apostrophe because Dr. Benedict, Stanley Benedict, is the one who came up with this test. Biuret is not like that. Biuret is a chemical that tests positive when these 2 chemicals are added, or biuret is a substance that tests positive when sodium hydroxide and copper sulfate are added to it. So it's called the biuret test. But that is not a person. So biuret is lowercase, no possessive. So biuret test. I don't want you calling the biuret, no biuret reagent. Okay? So no reagent. Reagents typically mean a number of chemicals mixed together that all have different functions, but they're in one, you put them all in at one time, in one solution. So like the Benedict's reagent is a number of different chemicals mixed together into a solution called Benedict's reagent, and then that's used. The biuret test really just uses 2 different types of chemicals. It uses sodium hydroxide and copper sulfate. So really, I don't want you to say biuret reagent. The way you'd refer to it would be something like the biuret test uses 2 chemicals, sodium hydroxide and copper sulfate. In the materials section, you are not, again, like in Lab 1, we're not going to use the red tide spec 20. We don't need cuvettes because they go along with the spec 20. And therefore, in the methods, we don't need steps 7 and 8. Those are about red tide spec 20 stuff. Okay? And additionally, what you need to add is a ruler, because when we do the biuret test, we're going to do the same 3 centimeters, 6 centimeters measurement that we did for the Benedict's. And there'll be 3 of the solution to be tested, and 3 more up to 6 centimeters of your sodium hydroxide. Then you're going to add 10 drops of the copper sulfate. 10%. So I'd also like you to add 1% copper sulfate. It says copper sulfate, just add the 1%. That's important. Wax pencil to make the marks and, label the tubes at the top. And then forceps are going to be used for the Sudan-3 test, which has a piece of paper that you're going to spot and let dry liquid on. So different substances in each of these little spots. You dry the liquid, then you spot it again to concentrate it. So it's on this filter paper. The filter paper goes in a dish. So that's your dish. The filter paper will go in the bottom of the dish. And the dish will have Sudan-3 stain in it, which will be red, and it will cover the, well, this is not really, but you understand what I mean. It will cover the filter paper. Then you'll leave the filter paper in the solution for a little while. Then you'll use the forceps to take it out. You will rinse it with tap water, and then you will look and see which spots, because they won't look like anything. You won't be able to see anything when you spot them on. They're all clearish substances. You'll see which ones look red. And the way they talk about it really is oranges. So the Sudan-3 itself is pale red. So if it looks pale red, that's just background staining. If you see accumulations of red, so there are orange. So there's some that might be light orange. Oh, that's why. There's some that might be light orange and some that are much darker orange. I could even do that. There's a light orange one. There's a darker orange-ish one. And so that just tells you the different concentrations, relative concentrations of the lipid content. And that would be the Sudan-3 test. Sudan is capitalized. It's actually named after the country. The Brits, when they're naming different stains, really like to name them after exotic locations in their view. So Sudan-3, we have like Congo red and things along those kinds of lines. And so that it really is it was named for the country because it was a pretty beautiful exotic color. So it's a red color, which will accumulate and turn the lipids orange. It actually, I take that back. Let's not phrase it like that. So the lipids are actually going to, the lipids sort of stay the same color. The deal is that the stain, the red stain, Sudan-3 stain, accumulates where lipids are present on the spot. And it does that because lipids are nonpolar. Sudan-3 stain is nonpolar. And that means they like each other. So basically it means they mix well. And they exclude the whereas like polar substance and nonpolar, these guys are not going to work together. So they would not. So you would not have the red stain sticking to a polar substance. Whereas with a lipid, which is a nonpolar substance, here, actually I want to draw it this way, you would have the lipid and the well, I want to draw an angry face, but I don't know how to do that. So anyway, these guys don't get together. So the only way that you have the red accumulating is if you have lipids, which are nonpolar. Then the hydrophobic interaction is the, and it doesn't need to be in quotes. It's called a hydrophobic interaction. Basically, that's them clinging together. And that's how the red happens. It doesn't change anything in red color. It just accumulates the red color or the red stain in one spot. Subtly different, but important. Okay, and you will basically, I think the rest of the information is fine in the lab. And of course that the instructions for that and specifically what you need to include from the textbook and whatnot are on the PDF that's online for Lab 2 instructions.